Good morning, everybody. So, we have discussed previously about microbial growth, growth media, microbial growth kinetics. Now, we are going to learn about culture system of microorganisms. We know there are two types of culture systems exist. One is open system and another is closed system. So in closed systems are also called batch cultures, in which nutrient supplies are not renewed or nor wasted removed. And exponential growth lasts for only a few generations and soon the stationary phase is reached. However, it is possible to grow microorganism in an open system, a system with constant environmental conditions maintained through continual provisions of nutrients and removal of wastes. And these conditions are met in the laboratory by continuous culture system. A microbial populations can be maintained in the exponential growth phase at a constant biomass concentrations for extended period in continuous culture system. So now we are going to discuss about more details about batch culture and continuous culture. So what is batch culture and what is its characteristics? In batch fermentations, all medium components are placed in the reactor at the start of the cultivations except for the atmospheric gases, acid or base for pH control and antifoaming agents. And there is a continuous change in the medium, nutrient medium concentrations over time, and the system remains unsteady. The microbial metabolites may be produced at a primary or secondary stage of the microbial cultivation period. Fermentations is terminated when either all the nutrients is exhausted or the desired concentration of products is achieved. This is actually the features of batch culture. Now, we will discuss about batch culture's advantage. Number one, it's reduce the risk of contaminations or cell mutations as the growth period is short. Number two, lower capital investment when compared to continuous process for the same bioreactor volume. And number three, more flexibility with varying products or biological system. And number four, higher raw material conversions levels and resulting from a controlled growth period. These are the advantages of the batch culture. Now, what is batch culture principles? A batch fermentation system is a closed system. At a time zero, an initial time, the sterilized nutrient solutions in the fermenter is inoculated with microorganism. That these are microorganisms which growth we are looking for, and incubations is allowed to process at a suitable temperature and gaseous environment for a suitable period. In the course of entire fermentations, nothing is added except oxygens in case of aerobic microorganism, and besides oxygen, an antifoaming agent, acid or base is used to control the pH. And the composition of the medium, the biomass concentrations and the metabolite concentrations generally change constantly as a result of metabolism of the cells. After the incubations of a sterile nutrient solutions with microorganisms and cultivation under physiological conditions, four typical phases of growth are observed. We are already learned in previous sections the growth phases like lag phase, like log phase, like stationary phase, and death phase. We are also learned each growth phases characteristics very well. So these typical four phases actually observed in batch culture. Now, what is the batch culture's applications? So it is batch culture is beneficial for construction of biomass like Baker's yeast system and primary metabolites like lactic acid, like citric acid, like acetic acid or ethanol productions. The batch culture ap- applied in food industries like organic acids are used as preservative or acidifiers such as lactic acid, citric acid, acetic acid. Alcohol beverage, in alcohol beverage like wine, beer, distilled spirit or brandy, whiskey and rum, and sweeteners like aspartate or amino acid used as flavoring agent. As for example, monosodium glutamate are the various product actually used in a day-to-day life, and that will be produced by only the batch culture cultivations. And here, like the applications, the wide applications in our day-to-day life. But besides the wide applications, it has some limitations too. It has some limitations too. So what are the limitations? Number one limitation is due to nutrient consumptions and waste buildup, microbes are exposed to constantly changing environmental conditions in a batch culture. It will change constantly. Environment will change constantly. And after reaching an end point, batch culture must be restarted. And in large bioreactor, it takes a long time to empty and to clean and to refill the reactor. And these are the limitations for batch cultures. And in batch cultures, the low productivity is consequence of the high downtime, non-production time, splint cleaning, sterilization, starting up another batch cultivations. These are all our limitations during two consecutive batches. And also, a buildup of toxic metabolites can sometimes inhibit the growth of the cells and product of the system. And these are the limitations you can face during the batch culture. Besides, it's a wide applications, but it has limitations. So now we are going to discuss about continuous culture systems. There are two major types of continuous culture system is used in day-to-day life or in industry or in the research purpose. Number one is chemostats and number two is turbidostat. So chemostat, here we have seen a chemostat images. This is the left side is the cartoon diagram. Okay, and you can see the fresh medium for reservoir, you can see the flow rate controller, you can see sterile air or other gas can be introduced into that part. Okay, and right hand images is the real images of the reactor actually. Okay, so a chemostat is constructed so that a sterile medium is fed into the culture vessels at the same rate as the media containing microorganism is removed. That is very important. You are filling the culture vessels and the sterile medium is fed into the culture vessels at the same rate the media containing the microorganism it will be removed. So that's why it is continuous system. And uh, we can say the population density is actually controlled by the concentration of limiting nutrient in the reservoir, and the growth rate is controlled by the dilution rate, and both the parameter are set by the experimenter can be set both the parameter. One is limiting nutrient and other is the growth rate. Okay, so chemostat. Now we'll learn previously what is chemostat and the important features of chemostat and what is the images of chemostat. Now what are the important points need to be know. Now one thing is need to learn very efficiently that chemostat and the concept of steady state, because it is a continuous system. So if it is a continuous system, there will be a steady state will be maintained. So here is a graph actually. There are dilution rate We have already learned that what is dilution rate. Dilution rate actually changes in limiting nutrient concentrations and also specific growth rate. How can it changes? Okay, so if you see during if you increase the dilution rate, the limiting nutrient concentrations, the bacterial biomass and specific growth rate and limiting nutrients changes, and how the changes will discuss now. The generation time decreases, that is the growth rate of growth increases. As the dilution rate increases, and the limited nutrient will be almost completely depleted under these balanced conditions. And if the dilution rate rises too high, the microorganism can actually be washed out of the culture vessels before reproducing, because the dilution rate is greater than the maximum growth rate. So this occurs because fewer microorganisms are present to consume the limiting nutrient. So we can say that the dilution rate is determined from the flow rate and the volume of the culture vessels Note that at a high dilution rate, growth cannot balance dilutions and the populations washed out. So it is also noted that although the population density, that is bacterial biomass, remain constant, and the concentration of the growth limiting nutrient remains near zero during steady state, the specific growth rate can vary over a wide range of time. So this is the relations in between chemostat principles and it maintains a steady state conditions. Now, another type of continuous culture system is turbidostat. So the second type of continuous culture system is turbidostat. So it has extra features compared to chemostat, that it has a photocells that measures the absorbance or turbidity of the culture in a growth vessels. If you see the cartoon diagram or right side of the slide, here you can see there is a photocells actually, and there is also a light source used, and this light actually sensed by the photocells, and that can be and the culture growth or turbidity or cell density is actually controlled over this by these photocells. So that means the flow rate of the media through the vessels is automatically regulated to maintain a predetermined turbidity or cell density. The turbidostat differs from ketostats in several ways. One of the main features is here is a light source and a photocells is used. And also the dilution rate in a turbidostat varies rather than remaining constant. And its culture medium contain all the nutrient in excesses. That is, none of the nutrient is limiting. The turbidostat operates best at high dilution rate, and the chemostat most stable and effective at lower dilution rate. Here like the comparison, here like the comparisons. Okay. So we are understand that what is chemostat. We are now understand that what is turbidostat and what is the difference between these two. So let's take a summary about chemostat and turbidostat and its comparisons. Chemostat, a system in which the chemical compositions is kept at a controlled level for the culture of microorganisms. And turbidostat, a continuous microbiological culture device which has a feedback between the turbidity of the culture vessels and the dilution rate. That means here feedback that is between the turbidity of the culture vessels and dilution rate will be controlled. Number 2 point, chemical composition of the medium is constant in case of turbidostat, but turbidity of the medium is constant in case of turbidostat. In chemostat, fresh medium is continuously added at the same rate as removal of the product. In turbidostat, fresh media is automatically added and maintain a constant turbidity. And very important point now is dilution rate. In case of chemostat, dilution rate remain constant. And in case of turbidostat, dilution rate varies. So, and also we can say in case of chemostat, processed within a limiting nutrient. But in turbidostat, there has no such limiting nutrient. So here like the comparisons, and chemostat and turbidostat nowadays used widely. So whenever you are using chemostat or turbidostat for continuous culture system, you need to remember those point here for making a culture. Now let's learn about application of continuous cultures. So we are already discussed about the what is called continuous culture, and what are the types of continuous cultures. Now the applications. Okay, so number one, continuous culture fermentations has been used for the production of single cell protein. Nowadays a single cell protein is very much important for research as well as industry purpose. Not only the single cell, but also to make organic solvents and starter cultures. Continuous culture fermentation is very, very important and applicative. Number 2 point, it has been used in the production of beer, this fermentation industry or food industry, fooded yeast, vinegars, bakery yeast, etc. And in the industrial productions of secondary metabolite such as antibiotic from a penicillium or a streptomyces species. Number 4, the continuous culture has been tested for L-lysine productions, and, and an important amino acid for metabolism. And this L-lysine producing C-glutamicum mutant B6. And also another application of continuous culture, it has been used in municipal waste treatment process. This is very, very important, and this is very, very much popular nowadays. It has continuous culture system has wide range of applications, but besides these applications, it has limitations too. So what are the limitations? Number one, in the long-term cultivation process, in the long-term continuous cultivation process, sterility maintenance can be tricky. It's very, very tricky, and downstream processing can prove challenging. It will face challenging in the downstream process. And we know in the fermentation process, we can get number of important byproduct in the downstream process. So it is very, very challenging in continuous culture of the downstream processing. Number two, controlling the production of some non-growth related products is not easy. Because of this, continuous culture often requires fed batch culture. So we will learn next the details about fed batch culture. What is fed batch culture? What are the features and what are the applications? The fed batch culture, and we'll then compare with continuous, fed batch, and batch. Okay. Point number three, the limitation point number three, as a result of viscosity and heterogeneous nature of the mixture, it may be challenging to maintain filamentous organism. And not only this, but also it has a limitation. If a faster growing strain overtakes the original product strain, it may be lost over time. This is very, very important, and these limitations having in continuous culture system. So during the previous slide, we get that fed batch culturing term. So now we need to cover what is fed batch culture and what is its features and what is its applications. So in simple word, fed batch culture is a modification to batch fermentations. In batch cultivations, nutrients are added aseptically, and it is a semi-open system, and the volume of the liquid culture in the bioreactor increase as the culture is systematically added. A fed batch culture is more productive, yield better with controlled sequential addition of nutrients, enables higher cell densities and prolonged product synthesis. So now we need to know the advantages of fed batch culture over batch culture. So what are the advantage of fed batch culture over batch culture? Number one, long-term synthesis of the product. For required long-term synthesis of product, you need to go with fed batch culture. By increasing the number of cells and thereby increasing the amount of product which is proportional to the concentration of the biomass, and greater efficiencies can be achieved in the fed batch culture. You will not get in batch cultures. Point number 3, increasing yield and productivity with controlled sequential additions of nutrient. Number 4, it allows the bioreactor to be used productions for non-profitable periods when it would normally be prepared for the next batch. So in conclusions, we can say in a fed batch process, the broth does not generally need to be removed during the entire fermentation process, but amount of limiting nutrient added determines the rate of reactions. So here we can see the graphical representations of the fed batch culture. Look at that, the biomass is compared with time period. If you see, there are, if you add substrate, if you substrate is faded, then look at that the biomass will be increased. Look at that the biomass increase, and substrate concentrations, whenever you start, it's become gradually decreases. Again you are fed with substrate, then again it will decreasing, but the biomass is continuously increase. So here we can get an idea that how substrate feed is important to get a continuous biomass or continuous byproduct from the cultures. So now we need to know about the applications of fed batch culture. So various product produced or attempt to be produced by fed batch culture techniques. Number 1, amino acid. This is very important for growth and metabolism. Amino acid like glutamic acid, amino acid like lysine, amino acid like tyrosine, tryptophan, alanine can be produced by fed batch culture. Antibiotic is very important. Candidine, candidexin, cephalosporin C, griseofulvin, penicillin, streptomycin, chlorotetracycline, tetracycline, all are can be produced in fed batch culture. Enzyme, as per example, enzyme, cellulase, galactosidase, beta-glucosidase, alpha-amylase, beta-amylase, galactosidase, all will be produced, all enzymes will be produced by fed batch culture. Solvent like acetone, butanol, glycerol 1,3-propanediol will be produced in fed batch culture. Vitamins, in case of vitamins, the riboflavin, vitamin B12 also produced in fed batch culture. And not only the product, but also microbial cells like bacteria cells, like yeast cells, as for example, Pseudomonas species, Pseudomonas candida boindi or Candida utilis. These are all are examples, but these are all are example of microorganism which can be grow, or microbial cell mass can be increased by fed batch culture technique. And besides microbial cells, animal cells culture and also some acetic acid, citric acid, gibberellins can be produced or can be used to produce by this technique. So now we are already discussed about batch culture, we are already discussed about fed batch culture and continuous culture. Let's have a comparative study. So in batch fermentations, all the medium components are placed in a reactor at the start of the cultivations. But in case of fed batch, a fed batch culture is a modifications to the batch fermentations in which nutrients are systematically added. And in continuous culture is a continuous process where nutrients are continually added to the bioreactor and the culture broth is removed at a safe time. This is the main principles and main comparative point, that in fed batch culture, all the medium components are placed in a reactor at the start of the cultivations. In fed batch culture, modification to the batch fermentation in which nutrients are systematically added, but in continuous cultures, continually added to the bioreactors of the culture broth. And also these cell metabolites or microorganisms can be removed at the same time. Number 2 point, fermentation is carried out in a closed system. But in case of fed batch, fermentation is carried out in semi-closed system. And in case of continuous culture, fermentation is carried out in an open system. So number 3 point is batch culture is a discontinuous process, whereas fed batch culture, you can say it's a semi-continuous process, and continuous culture is also, the name hence itself is significance, that is a continuous process. And the nutrients are supplied at the beginning of the cultivation in case of batch cultures without further addition later on. In case of fed batch culture, the nutrients are supplied at the beginning and later on added systematically during the process of cultivations. And in case of continuous culture, the nutrients are added continuously throughout the process of cultivations and the product form is continuously taken out. So so this idea is our clear that what is batch culture, what is fed batch culture, and what is continuous culture. Now, apart from fed batch and continuous culture, there is a special type of growth can be seen, and it is used industrially as well as in research. That's synchronous growth of bacteria. So what is the meaning of synchronous growth of bacteria? That means all the cells of a bacterial culture are same stage of their growth cycle is referred to as synchronous growth. If you look at the graph right side, it is a time versus number of viable cells. You will see a strial-like appearance. So what the significance is this? When bacterial cells grow, we also discussed in earlier as the growth curve. If observed under microscope, we have different stages of its reproductions. That is, some cells in elongated state, some cells in septum formation, divided of the cells, you are already discussed. So the cells and cell divisions will not be uniform. That means in a populations, some cells will in at the end of the division, some cells is the start of the division, some cells are the inner division. They are not uniform. But in synchronous cultures, if you do a synchronous culture, the bacterial cells will be on the uniform stage, all in a same stage. And the synchronous growth of a bacteria will have the growth curve as following graphs. That graph is depicted in the figure. The synchronous growth of a bacteria can be achieved. How can it be achieved? It can be achieved by growing the cells at suboptimal temperature. That means it equally required optimal temperature for growth 37 degrees Celsius, right? So you need to grow equally below the optimum temperature, below 37 degrees Celsius. So if you grow equally below 37 degrees Celsius, as for example 28 degrees Celsius, the culture will be synchronized. This type of culture will be useful. Why it is used? It is used in molecular biology or gene expressions or transformation studies. It's very, very important in case of research as well as recombinant DNA technology and in case of industrial purposes. And the last point we will discuss about dioxic growth curve. We will discuss about dioxic growth curve. So what is dioxic growth curve? Dioxic growth curve is a diphasic growth represented by the two growth curve intervened by a short lag phase produced by an organism utilizing two different substrates, one of which is glucose. When equally grow in a medium containing both glucose and lactose, it uses glucose preferentially until the glucose is exhausted. So you can understand this diphasic growth curve in the graph, where look at that it is here bacterial density plotted versus time. In a medium, you are adding glucose as well as lactose, but the bacteria will utilize first glucose. After utilization of the glucose, then again it will start to utilize lactose. But they need a time, and that is the short lag phase, because need to express some genes, need to express some protein or enzymes that will be help for utilizing of the second substrate is lactose. So that is actually called dioxic growth curve. So here it is written that after a short lag phase during which the bacterium synthesizes the enzyme needed for lactose is used, growth resumes with lactose as a carbon source. If this diphasic growth of the equal is plotted in respect to bacterial density against time, the two growth curves follow one after other. and that is intervened by a short lag phase to produce a dioxic or diphasic growth curve. So we are already talking about the enzyme. We are already talking about the enzymes that needed for utilize second substrate. The what is this? The enzyme needed for lactose is used actually beta galactosidase, and which split lactose into glucose and galactose. And the bacterium utilize glucose for the growth. So galactose can be utilized, but only after it is converted to glucose. So it has been demonstrated that equally growing in a medium containing both glucose and galactose produce a dioxic growth curve, as in case of glucose and lactose. Similar response has been found in case of other sugars such as arabinose. If you use arabinose, if you use maltose, if you use sorbitol, when they are used in combination with glucose by equally, each of these sugar is utilized only after glucose has been used up by the medium, and you will see a dioxic growth curve. And the cause of dioxic growth curve is complex. Is not completely understood till now, but it is considered that the catabolite repression of the glucose effect probably plays a part in it. And in catabolite repressions of the lack of peron of equally, glucose exerts an inhibitory effect on the transcription of the lac genes. You will learn this sort of gene expression and protein in later, but it is actually the gene expressions. And as a result, lactose utilization enzymes are not synthesized. Even if lactose is present in the medium, when glucose is completely consumed by the E. coli, bacteria are now competent to transcribe lac operon genes, resulting in a production of necessary enzyme that will help to metabolize lactose. And that is the principle of dioxic growth curve. So dioxic growth curve actually depend on the substrate and its gene expressions. So here it is the session's end. So thank you all.